Hello, my name is Marcin Ogrodniczuk. I'm currently a junior attending East Stroudsburg University, and today I'll be talking to you about my research on the expression of shikimate kinase as a potential target for novel antimicrobials. Now, before I dive into the details of our research, I'd like to give a short background on tuberculosis to help our viewers understand why our research is relevant. Tuberculosis is an upper respiratory infection that infected nearly 11 million people in 2017, which is far more than past years. This infection rate is primarily due to the fact that tuberculosis can exist in a variety of forms based on its level of resilience to antibiotics. This rising infection rate suggests there is a need for the development of novel antitubercular agents. Now, when searching for potential enzymatic pathways, it is vital that the pathway contain no mammalian analog. The shikimate pathway, shown here, is a 7-step metabolic route linking the metabolism of carbohydrates to the synthesis of essential cellular metabolites. This pathway is used by bacteria, fungi, and plants to synthesize aromatic compounds. This pathway has no mammalian analog, thus making it a suitable target for the development of novel antitubercular agents. Our research is focused on the 5th step of the shikimate pathway, shown here, which is the phosphorylation of shikimate to shikimate 3 phosphate. This process is catalyzed by our enzyme of interest, shikimate kinase, or in our case, Mycobacterium tuberculosis shikimate kinase. Below is the crystal structure of the enzyme, where you can see the conformationally flexible lid domain, the nucleotide binding domain, and the substrate binding domain. Our initial methods entailed a series of bacterial transformations using XL1 blue cells and DE3 gold cells. These were transformed using PET-20B plasmids with an ROAK gene inserted into the multiple cloning site. Our first transformation was deemed successful as we begin to see banding around 4100 base pairs, shown here. This is consistent with what we expected as it corresponds to the molecular weight of our PET-20B plasmid with an incorporated ROAK gene. This suggested that the XL blue cells were successfully transformed with the PET-20B plasmid. Our second transformation entailed us using DE3 gold cells. These cells acted as our expression hosts for shikimate kinase. The success of the protein expression was evaluated using SDS page analysis. Transformed DE3 gold cells were used to inoculate a series of lysogenic broths that were supplemented with both ampicillin and chlorophenicol. Finally, shikimate kinase synthesis was induced by the addition of 1 mole IPTG, and cell growth was monitored over 4 hours. Samples were collected every hour and an SDS page analysis was performed. The 4th hour sample was used to perform a TCA precipitation analysis and solubility test, shown here. There was a band of increasing predominance from T0 to T4 through the solubility test around 20 kDa. MTSK has a molecular weight of 19.6 kDa. This showed that the small-scale expression was successful. The bacterial culture was then upscaled for a large-scale expression and purification. Large-scale expression showed similar results to small-scale. We know here that the protein is being expressed in both soluble and inclusion bodies, as seen in the SDS page gel for insoluble and soluble. The protein also appears to be dimerized with a band around 40 kDa shown here. Subsequently, we purified the protein using nickel affinity chromatography. Altogether, we have successfully cloned the plasmid of interest, and our data shows that our protein is being expressed in both soluble and insoluble forms. We've also been able to purify shikimate kinase using nickel affinity chromatography. Our future studies will include the steady-state kinetic characterization of shikimate kinase and evaluating the effects of marine sponge metabolites like hindamine and derivatives of averone to determine their mode of inhibition and suitability for subsequent evaluations in the drug discovery and development pipeline. I would like to thank East Stroudsburg University Department of Chemistry and Biochemistry, as well as East Stroudsburg University College of Arts and Sciences. Thank you for your attention, and please stay safe.